A very good afternoon to everyone. Welcome to the post-lunch session of 5 days FDP on recent advances in biosimilars development. Now, the second speaker of the program is Dr. Utpal Mohan. He is currently the assistant professor in the Department of Medicinal Chemistry, Naipur, Kolkata. And he has done his PhD from Naipur, Mohali in the year 2009. And then he continued as a research associate there for an year before moving to Colorado State University in USA as a postdoctoral fellow. His research interests are development of synthetic riboswitches to suppress gene expression in case of various life-threatening diseases, development of deoxyribozymes which could degrade DNA, RNA of various oncogenes, generation of various novel protein coding sequences which could be further developed as biopharmaceuticals, suitability of G quadruplexes as gene regulation tool in cancer and other diseases, and the directed evaluation of therapeutic proteins for enhanced activity and least immunogenicity. So with this brief introduction, I request Dr. Utpal Mohan to deliver a talk on recombinant DNA technology and controlled gene expression methods for biosimilar production. Over to Dr. Utpal Mohan.
Thank you, Dr. Somachandran. At the onset, I would like to thank Director Sir, Dr. Ravichandran Sir, for giving me this opportunity to express my area of research in this biosimilar production. And so today I will be talking about recombinant DNA technology and the various ways of controlling the gene expression, which could be utilized for biologics also, as well as biosimilar production. So I won't be spending much time on this particular slide, the biosimilars, because Satish Pishi sir has already covered it very comprehensively. So I will just still read this out. The biosimilar is a biologic which is highly similar and has no clinical meaningful differences from the biologic, which is also known as original biologic or the reference product. Biologics, they are made with the same types of natural resources. If it is a like protein, same amino acids have to be there as the original medication they were compared to. And a biosimilar provides the same treatment benefits as the original biologic. So with this brief introduction, I will just talk about the various terms which are utilized to refer to biosimilars. These are follow-on biologics, follow-on protein products, biogenerics, and generic biologics. So these are various terms which are synonymous to the term biosimilar. So let's talk about small molecule drugs and biologics. So if you see the production of small molecule drugs and the biologics, so that makes it very clear why the protein biologics, the copy of the biologics are called biosimilars, and whereas the copy of an original small molecule drug is known as a generic drug, which is exact copy. Because if you see, these are all chemical medicines, they are made by the chemists, and they exactly follow the same recipe and it yields the same product every time. But biologics are complicated products. They are grown from living things. They must be manufactured under precise conditions and exacting steps to yield a consistent product. But still, they are highly sensitive to manufacturing conditions, and it is a biological process. So there are going to be differences which will make it little different from the original biologic. So that is why biosimilar is the appropriate word to use for such products. We will just see what is to look into this further deeply. If you start with the DNA is cloned into vector, so is the vector the same? A vector could be different. So when you use different vectors, many vectors give different properties to the product, and that may give slight differences in the product. So is it transferred to a cell? Yeah, or it is transferred to a cell. Is the recombinant cell line the same? So cell lines could be different. It doesn't have to be the same cell line used by the original biologic production. So again, there is a factor where differences can pop up. Then again, the fermentation, it goes through the fermentation. So is the fermentation process the same? Again, the biosimilar producing companies, they optimize the fermentation process in such a manner to bring down the cost, because overall a logic of producing biosimilars is bringing down the cost of the biosimilar as compared to the original biologic. So, and if you see the downstream processing, is the downstream processing always the same? No, it is not mandatory. It may differ because the biosimilar producer may use different ways to optimize its downstream process. So again, there is a point of difference here. So my major area of focus today of this talk would be various expression systems and how various genetic elements are utilized to affect the production of the protein product. So the therapeutic protein products, the biologics or biosimilars, they can be produced in microbial cells. It could be prokaryotic, eukaryotic. They could be cell lines, mammalian, avian, insect, and plants, or tissues derived from animal or plants. It is expected that this expression vector construct for the proposed product will code the exact amino acids. All the primary amino acid sequence will be same as the original product or the reference product. However, the smaller modifications, mostly in the post-translation modification, that are not expected to change the product performance, could be justified and should be explained by the producer or the sponsor. Possible differences between the chosen expression system, the host cell and the expression construct of the proposed product and that of the reference product should be very carefully considered, because what it will do is it affect the types of processes and product related substances. It also result in different kind of impurities and contaminants which may be present in the protein product. For example, the expression systems can have a significant effect on the types and extents of translation and post-translation modification that are imparted to the proposed product, which may introduce additional uncertainty into the demonstration that the proposed product is biosimilar to the reference product. So, by minimizing the differences between the proposed product and reference product, expression systems to the extent possible can enhance the likelihood of producing a biosimilar protein product. And we can use different expression systems by case basis. Following are the factors which determine the type of expression systems which could be used to produce recombinant proteins: the time spent in expressing the protein, ease of handling the expression system, amount of protein needed, mass of the protein, type of post-translation modifications, a number of disulfide bonds, and other type of modification, destination of the expressed protein, where you want the actual protein to land after the product is formed. So the process of expressing a recombinant protein in expression system will need these components. First would be the identification of the gene that encodes the protein of interest. It could be a protein like a growth hormone, like this any growth hormone, or like insulin product, or a monoclonal antibody. But you need to have a gene sequence. You have to have the knowledge about the sequence of the gene which you are going to produce. Then you need to generate the complementary DNA from the respective mRNA if you are dealing with a eukaryotic therapeutic protein. So selection of a suitable expression vector to insert the gene sequence. Then you have to select suitable system that can express the vector. That means the host expression system, which will support or will be compatible with this vector. And then the appropriate scaling and screening methods. These are the factors which are needed. So general overview of the expression systems will be, first of all, definitely you have to know what biosimilar you are going to target. You need to know the full length cDNA sequence. Then you have to insert it into expression vectors. Then you have to clone into suitable expression system. Various expression system I am going to explain in the following slides. Screen for positive clones, select the best construct or expression host, then go for either mammalian cell expression, bacterial, yeast, insect, or plant cells. So then the isolation and purification of the protein of interest and the downstream processing thereafter. But I will be restricting myself from the recombinant DNA aspect. Bacterial protein expression systems, there are many protein expression systems So one of the most commonly used protein expression systems is E. coli system. So why it is so popular and so much in use is because it is a rapid and simple system for expressing recombinant proteins, and the doubling time of E. coli is 20 minutes. So the media required to culture them are not expensive, and the methods adapted to scale up bioproduction are pretty straightforward, not like the mammalian cell fermentation methods, which are pretty complicated and have very high cost requirements. The most widely used host system is E. coli, since there is ample knowledge about its genetics, genome sequence, and physiology. And the manipulation of the genes within the vector is easy, and it also allows to high densities and is suitable for large-scale fermentation. I would like to put a point right here that even if you are going to clone it in mammalian vectors or other vectors, but the first thing is you always, any gene, you have to clone it in a bacterial system. You try to make the clone in the bacterial system. Once that is ready, you subclone it from there to the mammalian vector or plant vector or insect vector, whichever expression vector you have chosen for your particular biosimilar. However, the cell wall of E. coli contains toxic pyrogens, and the expressed proteins may be extensively tested before use. So you need to keep this consideration in mind before going ahead with this particular expression system for your particular biosimilar. What are the features? So the first feature would be the low-cost culture methods. Flexible systems can carry plasmids with multiple promoters, tags, and size, and that's why you utilize this cloning system or this vector system for cloning your gene. The first thing you will do is clone it in E. coli. Once E. coli vector or bacterial vectors, prokaryotic vector. Once you are done with that, you will subclone it into various other vectors according to the choice of your expression system, and they are easy to scale up and produce high yield of proteins. So this is the overview of the bacterial expression system. So once the this particular gene has been identified, you will be cloning it in a bacterial expression vector. Once it is cloned and it is sequenced and it is verified that the gene of interest is in frame, it is very important for this gene product to be converted into protein, because if it is not in frame, the gene will never be converted into a protein product. So it has to be sequenced, it has to be checked here itself at the second point. So once you select it and then when you identify the positive clone, you sequence it. Once that step is done, you then go ahead and you add it in the you scale up the transformation isolation of protein of interest. So you isolate the protein and you go ahead with the downstream processes. So moving to the next protein expression system, which is yeast protein expression system, specifically the Saccharomyces cerevisiae. This is a highly developed genetic system. It is again easy to use. It has again reduced time input as compared to mammalian expression systems, and costs have made cerevisiae and reduced costs have made this particular system attractive organism for expression and production of these therapeutic proteins. Yeasts are able to carry specifically designed plasmids, and this ability is valuable in protein expression system. So this plasmid use consists of size that can be used to insert the gene of interest, which is common in every expression system. Transformation of yeast with the plasmid produces the desired protein and can be appropriately scaled up. What are the features of this particular expression system? They are again low-cost culture methods suitable for both intracellular and secretory proteins. One important difference from the prokaryotic system here is it provides post-translational glycosylation of proteins, and many of the eukaryotic proteins, for their actual activity, they need post-translational modification. So that is why bacterial systems are sometimes lag behind as compared to the insect expression system or yeast expression system or mammalian or plant expression systems. So because they don't have, the bacterial system don't have the sophisticated post-translational modification in those cells. So that is why the therapeutic proteins, which are generally of human origin, they need complex post-translational modifications. Sometimes, not every time, because if you see insulin is produced in E. coli, and there are many examples of biosimilars and biologics which are produced in E. coli, and they are marketed from that particular production system. So coming to the overview, again the same flow chart is here. We have to clone it in the expression vector, the yeast expression vector. We have to transform it into the yeast, and once you select it, you scale up the transformation and isolation of the protein of interest. So it follows the same flow chart as the bacterial expression system. Moving on to another expression system, which is insect cell expression system. These cells are derived from insect, Spodoptera, Sogyroperda, SF9 and SF21. And they are frequently used as recombinant protein expression systems, and we also call them baculovirus system, because baculovirus is a lytic virus which infects these insect cells. So it routinely this virus is routinely amplified in these insect cells, SF9 and SF21 insect cells belonging to this Lepidoptera family. It is non-infectious in vertebrates, and its promoters are inactive in mammalian cells. So it is very easily used for expression of many proteins, many therapeutic proteins also, because this also offers some post-translation modifications as compared to the bacterial expression system. Recombinant protein is highly expressed during the last phases of lytic cycle before cell lysis. These are the important features suitable to generate both cytoplasmic and secreted proteins. There are disulfide bond requirement in many of the proteins. If you talk about the antibodies and many other proteins for their efficacy, so they are these particular insect cells can provide disulfide bonds, and it also provides majority of post-translational modifications which are generally found in mammalian cells. So insect cells are easy to handle as compared to the mammalian cells. So if your biosimilar doesn't have very specific requirement of particular post-translational modifications, this particular expression system could be considered also. So this is the same. I won't be spending much time on this particular slide. It follows the same. You have to identify the gene, clone it in a vector, transfect it. So once you co-transfection of insect cell lines, production of high titer. So basically you are transfecting two things. One is the plasmid with the viral gene, and one is the plasmid or expression vector having a gene of interest. So then it will help in production of high titer recombinant virus stock, and eventually the insect cells will be producing the recombinant virus stock, and isolation of protein of interest will be possible. So moving on to another expression system, which is mammalian cell expression systems. So generally for monoclonal antibodies and other therapeutic proteins, when we utilize the mammalian expression system, the Chinese hamster ovary and human embryonic kidney cells 293 are generally utilized. The main challenge of using these cells for express is the reduced efficiency and levels of the protein expressed. So there is this particular disadvantage, but again, because the need of specific modifications on the protein surface, which makes the protein or biosimilar active, makes the pharmaceutical company to go for these expression systems. So however, cell lines such as HEC293 and CH2 have been developed as efficient, transient, and stable expression systems respectively. So these HEC293 cells are transiently transfected using liposomes. So there are two options. One is like you can go for transient transfection, another is you can go for a stable transfection. So basically in the stable transfection, it will be incorporated into the chromosomal DNA. So CHO cells are commonly used to stably express large quantities of recombinant proteins. They involve infection of DHFR deficient CHO cells with the gene of interest and a DHFR selection cassette. The transfected cells are then screened in the presence of methotrexate for DHFR selection to obtain stably transfected cell pools, and further the selection expansion. So this is basically the process for selection of stable cell lines. So again, this similar flow chart is there. You have to know the gene sequence. You have to clone it in the vector. You have to send it to the cells, and then if you are going for a transient expression, you have to screen an expansion of clones for a week. If you are going for a stable expression, you need to screen it for 2 to 3 months under the antibiotic pressure. So there are another expression systems which are coming up these days. So monoclonal antibodies are also being produced in the plant expression systems. Unlike other systems, plants have some additional benefits. So what they give you is like they have like they can carry out complex post-translational modifications which are necessary for the function of many biopharmaceuticals. So plants stand equal with the mammalian systems when we talk about post-translational modifications. So, and unlike mammalian systems, which require large investment and operating costs, these systems are not requiring that much expensive cell culture facilities and bioreactors, because you can grow it, you can send it inside a plant cell, and that plant will be the producer of your biosimilar. So advances are being made further to see whether this could be a very reliable expression system, and whether it could be whether the technology could be transferred, and whether it could be produced in a large scale in an industry. But this is definitely a very exciting expression system. So while I'm talking about these expression systems, I need to talk about some of the vectors which are largely utilized for monoclonal antibody-based biologic production. So these are known as tandem vectors. Why they are called tandem vectors? Because they are equipped to express containing heavy chain cDNA and light chain cDNA in tandem. So you have a single vector which has internal ribosome entry sequence, followed by a heavy chain cDNA, followed by another is cistron. So basically these are bicistronic vectors. They have two cistrons. You are having heavy chain cDNA, also the light chain cDNA. Earlier people used to have two vectors for expressing this heavy chain cDNA and light chain cDNA. Now people are trying to, researchers are recommending the tandem vectors, and industries are also utilizing these tandem vectors for the production of monoclonal antibodies. And in the construction of these tandem vectors, the probability of promoter interference should be taken into account, because this leads to the unstable RNA product. So for this reason, there are two unrelated promoters, CMV-MIE of human cytomegalovirus and the human translation elongation factor, the 1-alpha gene promoter. They are utilized to avoid this problem. So few considerations I would like to talk about here for the MAB biosimilars. The growing demand for monoclonal antibodies has led to the investigation of several plasmid vector designs. So this tandem vectors are one of those. For instance, different signal peptide sequences are known to strongly affect the protein secretion. Therefore, signal peptides for the heavy chain and the light chain must be carefully selected to ensure an efficient secretion of properly folded and assembled antibodies. So these are the considerations which are very crucial while going for monoclonal antibody based biologics or biosimilar production. And another important aspect is the regulation of light chain and heavy chain expression ratio. Higher light chain over heavy chain expression proved to be beneficial for protein folding and aggregation reduction. So it has been reported that if you go for higher LC light chain is to heavy chain expression, so then you will be seeing lesser protein folding and aggregation issues. And then there is another concept of IRES, internal ribosome entry site, which allows the expression of more than one gene in a single transcript by creating cap independent ribosome binding sites. So IRES are these are the internal ribosome entry sites which allow the translation of a product. And because eukaryotic expression system, in the eukaryotic expression system, the mRNA needs a 7-methyl guanosine cap at the 5' end. But if the IRES sites allow and give that independence, because if you have IRS, then the 5' cap requirement is no longer there. So that is why these IRS are very much used for production of biosimilars, and creating bicistronic vectors. IRS are frequently used. What are the factors we need to consider? So these could be codon usage and codon bias. So codon usage, as we all know, these are the problems not only for biosimilar. These are the problems for expression. These are the problems for expressing any gene and for any biologic or any biosimilar. So you have to optimize the gene based on the expression system. So based on the because there is a degeneration in the genetic code, every bacteria or every expression host, like it could be insect, it could be bacteria, it could be yeast, plant, they have a different codon preference for coding a particular amino acid. So that codon bias has to be kept in mind while you are choosing the sequence which you are going to clone in a expression vector. If it is a codon optimized sequence, then only chances are high that you will get a good expression. Otherwise, there are many instances when like if you have not kept this thing in your mind, optimizing the gene based on the codon usage, then it will give you lesser yields of the final product. So there are many tools like IDT, integrated DNA technology. They have their oligoanalyzer tool. So they also have a tool which gives you the to optimize the gene. If you input your gene sequence, it will optimize it based on the host you are interested in expressing your protein. And similarly, the protein folding, you have to keep you have to consider protein folding. So inclusion bodies are also one of the considerations here. So formation of inclusion bodies will eventually, they are the misfolded protein, so you need to take in account that particular point also. And then post-translation modification, I have already mentioned the post-translation modifications. What we have to see is like what kind of protein I am dealing with. So if this protein, whether this protein needs very sophisticated or very complicated translation, post-translation modification. If not, then you go for an easy expression system. You go for E. coli first. First, see can you do it with the E. coli system or the bacterial system, because that is very easy to manipulate, handle, cost efficient, effective, and many benefits are attached to that particular expression system. So you keep on coming up. If you know you have a higher post-translational requirement, you go up. Can yeast manage those? Can insect cells manage those? Can your like if not insect cells, then you have to come to the mammalian cells or the plant cells. So plant cells is a growing up field, plant expression system, but definitely it has very high hope. We all hope that this will be very successful expression system, and if that is the case, then the biosimilar field will be very much benefited. And then coming to the stability of mRNA. So stability of mRNA, if you think of it, the moment the mRNA is synthesized, it has a small half-life. It doesn't stay for too long. So like few minutes maybe. So what happens is, if the stability is not much, so the product, the protein product will be, the yield of the protein product will be affected based on the stability of mRNA. There are ways to increase the stability of mRNA. I will mention a couple of examples in my next slides. And then there comes the promoter strength and the inducers. So what kind of promoter strength you are looking for, and what inducers. If it is a very high promoter strength, then also many a times, because of the very high expression system, a very high overexpression, what will happen is there is a increased inclusion body formation. And what kind of inducer you are you want to use, if you are using the bacterial systems or yeast systems. So because you are an industry and you are thinking in that aspect, the cost reduction, so you will not go for inducers like IPTG, which are very costly inducers. So you need to look for, so every biopharmaceutical company or the company dealing in biologics look for their own vector system. They make their own vector system so that they customize their promoter inducer based on their requirements. So I'm almost at the end of my talk. So I've just mentioned about the mRNA stabilization. I will just further add few new points about this particular UTR. So there is an outer membrane protein which is found in the E. coli. So it has been found that this particular UTR of this OMP attains a very specific secondary structure. So what happens is when you utilize this OMP 5' UTR, it stabilizes your overall RNA, the RNA, mRNA, and of the biosimilar or the biologic. So that gives enough time for increased protein production from that single mRNA molecule. Okay, so that will eventually lead to increase in the yield of your biosimilar. So like this, it can prolong the cytoplasmic lifetimes of a variety of messages to which it is fused. So it has been seen widely that when you take this 5' UTR sequence and clone it in front of any gene, any bacterial gene, this is mostly effective in the bacterial system. So if you are utilizing the bacterial expression system, this is a good system, good UTR, which you could fuse it with the gene of your biosimilar. Once you fuse it, this 5' UTR of OMP, outer membrane protein A, will stabilize your mRNA. What will happen is actually the secondary loop structure avoids the RNase-based degradation. It avoids the RNase-based degradation, and that is why the accessibility of the ribosome binding site to the RNase or the degradation stops. So that is why the degradation is prolonged and the mRNA stays for a longer time. The half-life is increased, and that leads to increased biosimilar product. So there are two different domains which have been identified in this particular UTR, the untranslated region. You all are aware the untranslated regions are that part of the mRNA which will not be translated into protein. Okay, so it is, I'm talking about the 5' part on the left hand side of the gene. So then the first one is the domain is responsible for stabilizing effects. One is a 5' terminal stem loop, and the second one is the single stranded RNA segment that contains a ribosome binding site highly complementary to the 16S ribosomal RNA. So this is a very useful trick we can utilize in expression vectors to increase the mRNA stability, which can lead to increase in our product. So I have come to the end of my talk. So I would like to summarize that we have talked about the various aspects of biology for which biosimilar blend needs to be considered. So we have to take into account all these factors before choosing starting the first thing, the gene sequence optimization, codon optimization, then which vector to choose, which expression system to choose. These are all necessary considerations which will have a very huge significant effect on the final product. So various expression systems are available which could be selected based on the above mentioned considerations. Definitely gene expression enhancement and mRNA stability consideration could be very well utilized to increase the yield of the biosimilar protein product to bring down the final cost. So I would end my talk with this particular slide. Thank you very much for your kind attention. Yeah.
Thank you, Dr. Utpal, for your excellent talk on the genetic techniques which are involved in this biosimilar development, especially the recombinant DNA technique. And, I request audience, if you have any questions, there's a chat box available, so you can post your questions there. Dr. Utpal, one question. Sir, can you please elaborate a bit on plant-based biosimilar products?
Yeah, plant-based biosimilar products, mostly what people are trying to do is they are utilizing people have just shown that they are able to utilize the agrobacterium tumefacial based expression system to send the light chain and the heavy chain of the monoclonal antibodies, and they are cloning it into agrobacterium vectors. Then they utilize this agrobacterium system for transfixion of the plant cells. Once these heavy chain and light chain enters the plant cell, the production begins just like any other protein. Only thing you have to again keep in mind is the codon optimization according to the plant cell codon usage. So once you do that, people have seen that they are getting enhanced protein production. And as I just said about the outer membrane protein mRNA, so similarly, there are many 5' UTR elements in the eukaryotic system, which when added to a particular RNA message of a biosimilar or biologic product, it increases the stability of the overall transcript.
Yeah, thank you.
But I'm very hopeful about this plant expression systems, and I feel that in coming days, the biosimilars, we can make biosimilars at an industrial level in plant cells also. So I'm pretty hopeful, because that is why it is very useful. Is you don't have the issues of having like sterility issues are not there. There are many other advantages which are associated with the plant-based biosimilar production or the recombinant protein production, which are associated with all the mammalian expression. But at the same time, the plant expression system will give you the very sophisticated or very complicated post-translation modifications, which are only possible in plant cells if you are avoiding the mammalian cells.
So thank you. One more question. Is it possible to make recombinant insulin biosimilar with modified amino acid in both alpha and beta chains?
Yeah, see, as Satish Bishay sir said, as long as the biosimilar is, you can't change the original. If you change the primary sequence and it affects the biologic the activity, then it is no longer a biologic or biosimilar. It has to be a copy of the original product. When you talk about function, it has to do the same effect. But if you consider that, okay, we have added few amino acids, it is adding the stability, okay, but it is not, and it is helping in the secretion also, but it is not at all affecting the efficacy or any other toxicological effects or like preclinical testing. If it is not having any other issues, then it is just fine. You can do that, and people do that often. Primary sequence is generally not tinkered with.
Another question is, can we go for receptor oriented biosimilars?
Receptor oriented biosimilars. See, yeah, I would like to, that's a very good question.
Usually ligand oriented research what we are doing, right? Yeah, but this they're asking whether receptor oriented. Usually many things will act on receptors, so we can expect that.
Actually, and see, one thing I would like to make clear here, that most of the people, when they talk about biosimilars, they feel that the monoclonal antibodies are the biosimilars. It could be any protein. So as long as your protein, which is your receptor protein, is having a therapeutic effect, okay, so it will come under, if there is a original biologic based on a receptor protein, and then if you are making the same thing after the patent expiry, then it will be considered as a biosimilar if it meets the other requirements, definitely. So what I am trying to say here is, like, not only the monoclonal antibodies, anything, any enzyme, any like protein, therapeutic protein like insulin, growth factors, they all are biologics. When the patent expire, you can go for, as an entrepreneur, you can think of producing biosimilars, definitely.
Yeah. Which is the most widely used or accepted vector system for biosimilar production?
Okay, that is a very difficult question to answer because the first of all the companies won't these are the classified information of the all the companies. So what people generally do is they take elements like the promoter element. If you're talking about mammalian expression system, the CMV promoters are mostly used. So, and if you talk about like bacterial expression system has lot of flexibility, so you can't pinpoint what particular vector the industry is using. It is very difficult to know, but there are so many vectors available. We know that there are lac promoter based vectors, there are promoters with T7 promoters. There are very different types of bacterial promoters available, and similarly eukaryotic vectors are also there. There are a lot of options, but what you have to consider here is recently, like 3-4 years back, there was a call for proposal from the biopharmaceuticals from the government of India, where they were asking for researchers to come up with novel expression systems, novel expression vector design. Why they are asking this? Because we can't utilize a known expression vector which is already patented by a foreign company. So to facilitate the Indian biosimilar industry, government has given this call in the biopharmaceutical call. So they wanted researchers to come up with novel vector system, vector design, which will help in taking care of most of the problems we are facing, like secretion, like increasing the mRNA stability. All you can keep on adding various properties, and you can convert it into a circular plasmid, and then you can see whether the system is viable, and whatever you have started you want to get from it, is it delivering or not. So it is, you can't pinpoint a particular vector system because it will not be known to you. But there are known commercial vector, but those are all patented. You have to you have to tinker it with to make your own vector, and you have to look into what are your own requirements, where you want to cut costs. So these are the things. So like I just said that Omp A. So Omp A 5' UTR. So now what there is a company now which has started making vectors which provides bacterial vectors which provides Omp A 5' UTR already in it. So vector is already packed with Omp A 5' UTR. You just have to clone your gene on the downstream side of the Omp A 5' UTR, and it will give you increased protein product. So that is what the status is currently. So you have to know the elements, then you try to make your own vector, and that will be the best solution, I feel.
Yeah, thank you, Dr. Utpal, for spending your valuable time and sharing your expertise to all the
Thank you.
participants. I think they have gathered some knowledge on this recombinant DNA technology. Yeah, thank you. And I thank all the participants also for joining this post-lunch session, the second session by Dr. Utpal Mohan. And we will meet tomorrow at 10 a.m. for the second day event.
Thank you very much. Thank you.
Yeah, thank you, Ananda.
Sir.